All right, everyone, welcome back. We are now nearing the end of chapter 17, but we've got just a couple of pretty big topics left. One of them will be in this lecture, one in the next. So today we are going to talk about leukocytes, white blood cells, but at the same time, we are also going to jump ahead and cover a little bit of chapter 21, the immune system. So make sure that you have at least read the parts of chapter 21 that are in your outline within the chapter 17 blood outline. So as always, let's go ahead and start with our not really attendance questions. First, why does someone with type A blood have anti-B antibodies? And second, how do antibodies recognize foreign antigens? Go ahead and try to answer those. All right, hopefully you've done that, so let's see what the correct answers are. First, why does someone with type A blood have anti-B antibodies? Well, from our blood group and blood typing lecture and our lab, remember, whatever the blood type is, that tells us the antigens that are on the surface of those erythrocytes. And the way our immune system works, in part, is by using antibodies to find something that is out of place. So anti-B antibodies are looking for B antigens, which someone with type A blood should never have in their body. Which brings us to the next question. How do antibodies recognize foreign antigens, or how do they know what should or shouldn't be in the body? It's all about shape. Pretty much any time we talk about any protein, it's going to be about shape. Now, the way antibodies work is they are shaped to fit things that do not belong in the body. There are some antibodies that are shaped to fit B antigens. That way, if a B antigen ever does show up in the body, then these anti-B antibodies will stick to it and cause some reactions that we'll see later today. So now, so far, we have talked about blood in general. We've talked about plasma. We asked what formed elements are, and then we started talking about formed elements with erythrocytes. Now we are talking about white blood cells, which are also called leukocytes. That's what we will call them now, are leukocytes. Leuko means white, cyte means cell, so these are white cells, white blood cells. And white blood cells, there's a bunch of different types, but there are some things that they all have in common. And one of those things is they have amoeboid movement. Now, back in high school, or if you took a biology class, I'm sure you talked about amoebas. Amoebas are single-celled organisms, and they kind of crawl around by extending parts of the cell. And when they do that, it allows them to either approach or avoid something. So leukocytes behave like amoeba in the way that they move. They have amoeboid movement. And they exhibit something called positive chemotaxis. Chemotaxis just means movement in response to some chemical. Positive chemotaxis means they respond by moving towards a certain chemical. Negative chemotaxis would have meant they moved away from it. But leukocytes, white blood cells, move towards certain chemicals. Now, the normal value of leukocytes in the blood is about 4,800 to 10,800 cells per cubic millimeter. If there are fewer than that, we learned a term in lab called leukopenia. If there are more than that, then we have something called leukocytosis. And leukocytosis can be a diagnostic. For one thing, it could represent something like infection, because we're going to see today leukocytes are a big chunk of our immune system. So when our body is infected with something, we mount a defense by raising the number of leukocytes. So it would be higher. Leukocytosis can also represent certain types of blood tumors or something like that we'll talk about a little later. Now, as long as you've already done the blood lab, well, we have learned about granulocytes and agranulocytes, but I want to recap it here just in case you're watching this before lab. When we look at leukocytes under a microscope, some of them are filled with these little, almost like polka dots. These are certain proteins called granules. Not all leukocytes have visible granules. The ones that do, we call granulocytes. The ones that do not, we call agranulocytes. A meaning without. So granulocytes have granules that are visible. Agranulocytes do not have granules that are visible. Now, we talked in lab also about never let monkeys eat bananas. When we learn the names of leukocytes in a moment, this is going to be a mnemonic that lets us know the order of leukocytes from most abundant, beginning with N, to least abundant, beginning with B. So here are the leukocytes. And over on the left, we have something called a neutrophil. Neutrophils, when we look at them under a microscope, down here it says they have pale red and blue cytoplasmic granules. Now that's true, but when we look at them through a microscope, it's really hard to distinguish the kind of pale red and blue granules. They kind of blend together, and neutrophils tend to be kind of a pink color or sometimes an orange salmon color. So they kind of look pink or salmon color under a microscope. And they have multi-lobed nucleus. That is the thing that's most distinguishing about neutrophils. And this one, when we look at it, we see that there are 3 lobes to his nucleus. Here's a big one, here's a big one, here's a big one, and they are connected. It's one nucleus. It's not 3 different nuclei. It is one nucleus. They are all connected. Sometimes neutrophils will have 4 lobes, sometimes 5 lobes, and the shapes of those lobes will be dramatically different from one neutrophil to another. So another name for a neutrophil, we won't use it in this class, but you might see it later on down the road in other classes. Sometimes they are called polymorphonuclear leukocytes. Polymorphonuclear leukocytes. And that just means that the nucleus can look dramatically different across different neutrophils. Now, what do neutrophils do? These are the bacterious layers. They go after bacterial infections. They release substances from those cytoplasmic granules that help fight bacteria. Next, eosinophils. Eosinophils can at first resemble neutrophils. They have a multi-lobed nucleus, but their nucleus typically only ever has 2 lobes. I have seen a neutrophil with 3 lobes. The third was not very developed. So in general, we say they are bilobed. They have 2 lobes, and they are, again, connected by a string. This is not 2 nuclei. This is a single nucleus with 2 lobes. When we look at them under a microscope, the easiest way to distinguish it from a neutrophil is the color of the granules. They are very bright red. Very bright red. And if you're a comic book fan, I like to say the eosinophils look like Spider-Man or Deadpool. If you are familiar with either of those characters, this is what this looks like. Now, eosinophils, their primary function is to fight off parasitic worms. So if someone has a tapeworm, for example, eosinophil count will be elevated. Now, even though that's their function, they are related to certain types of asthma. Now, this is not a function of eosinophils, it's kind of a side effect. Eosinophils, when they're fighting parasites, they release substances from these granules. And some people are very sensitive to those granules, and it causes them to have asthma attacks. Sometimes when you're watching TV and you see one of those asthma medication commercials come on, you might hear them say something like, "This is to treat," or "This is not to treat eosinophilic asthma." That's what it's talking about. Next, basophils. Basophils, when you look at them under a microscope, I think they look like they are out of focus because you really can't see anything going on. But what's actually happening is they just have a lot of really, really dark staining granules. They're dark blue, dark purple, almost black. And there are so many of them, you do not see its nucleus. There is a nucleus present, and it's 2 or 3 lobes, usually 2 lobes like we have over here with the eosinophil. If you look really hard, you can see that nucleus in here. Usually in a microscope, you cannot. So it looks like it has no nucleus, or it looks like the nucleus is taking up the entire cell. That's not what's actually happening. Now, basophils, they release substances involved with inflammation called histamine and heparin. Histamine and heparin are inflammation mediators. And if you have allergies, basophils are in part to thank for that, because the histamine causes inflammation a lot of times either in the respiratory tract or in the sinuses of the nasal cavity. So that's why you take antihistamines to get rid of that. So those are the granulocytes: neutrophils, eosinophils, basophils. The next two are agranulocytes. They do not have visible cytoplasmic granules. The first one is a lymphocyte. Lymphocytes, now, when we look at them under a microscope, they can at first resemble basophils because they have this big, really dark staining thing, but actually this is its nucleus. It has a very big, mostly round-shaped nucleus. The way to differentiate it from a basophil is it will have a very small halo of pale blue cytoplasm with no granules in it. Lymphocytes are our primary immune response. And when we talk about the immune system in just a few minutes, really what we're going to be talking about are lymphocytes. These direct our adaptive immune system. And lastly, monocytes. Monocytes can also resemble lymphocytes and basophils. But the difference between a monocyte and a lymphocyte is the nucleus. It is also very, very big, but where the lymphocyte's nucleus is mostly round, a monocyte's nucleus is U-shaped or kidney bean-shaped. And we can see quite a bit of that pale blue cytoplasm with no visible granules. Monocytes, while we do find them circulating in the bloodstream quite a bit, they will typically leave the bloodstream, go out, and move through the tissues of the body. And when they do that, they change shape a little bit, and we call them macrophage. A macrophage, all that means is big eater. Macro, big, phage, eat. So these guys wander through the tissues eating things that should not be there in a process called phagocytosis. So we have talked about production of blood cells, the formed elements, and we called that hematopoiesis. We said if hematopoiesis was specifically making red blood cells, we called that erythropoiesis. But now we're going to talk about when we're making white blood cells, when we're making leukocytes. That process is called leukopoiesis. Leuko, white, poiesis, formation of. So making white blood cells is called leukopoiesis. Remember, erythropoiesis was directed by erythropoietin, EPO, from the kidneys. Leukopoiesis is stimulated by interleukins, which are chemicals released from some of the white blood cells, typically when there is some sort of foreign pathogen detected, and colony stimulating factors. So interleukins and colony stimulating factors. We also talked about something called thymosin back at the end of the endocrine system. Thymosin, it doesn't really stimulate leukopoiesis, but it stimulates development of certain types of white blood cells. Leukopoiesis, like all hematopoiesis, takes place in the red bone marrow. And then once those cells are made, some of them will actually move to other locations. Specifically, we're going to be talking about the thymus, but some of them will also go to our lymph nodes. So here is the process of leukopoiesis. Just as with the erythrocytes, we begin with a hematopoietic stem cell, a hemocytoblast. But if we're going to be making white blood cells, the process is a little bit different. And since we have several different types of white blood cells, there are several different paths. When the hemocytoblast divides, if we're going to be making white blood cells, we produce two cells. One of them remains a hemocytoblast. The other either becomes a myeloid stem cell or a lymphoid stem cell. Let's look at lymphoid stem cells first because there's less going on. Lymphoid stem cells divide to produce either B lymphocytes, B cells, or T lymphocytes, T cells. So that's what we have down here. Don't worry about the intermediate. Just know hemocytoblast, lymphoid stem cell, and then either B or T lymphocytes. We can see that B or T lymphocytes can become other things also. We will talk about that when we get to the immune system. The other thing hemocytoblasts can do, if they're going to become something other than a B or T cell, is divide. One cell remains a hemocytoblast, one cell becomes a myeloid stem cell. Myeloid stem cells then go through development. You do not need to know all of these intermediates. Myeloid stem cells divide to produce either eosinophils, basophils, neutrophils, or monocytes. Monocytes, as we said, can go through another change and become a macrophage out in the tissue. So again, all of these leukocytes, all of these white blood cells came from a hemocytoblast, which is also where erythrocytes, red blood cells, came from. Now, things can go wrong with leukocytes just like any other part of our body. There's lots of different disorders. We're going to cover a couple of them. One that most people are familiar with is leukemia. And again, like we keep running into, leukemia is not a disorder. It is a class of disorder. And different leukemias have different symptoms, different results. But leukemia in general is a cancer having to do with white blood cells, leukocytes. Normally, but not always, normally it is to do with one of these developmental pathways along the way. So you could have something like a leukemia involved with B cells, or a leukemia involved with T cells, or a leukemia involved with lymphoid cells, which would affect both B and T cells. Another disorder, infectious mononucleosis. This is sometimes just called mono. In the past, I don't know if it still is, but it was common for people to call it the kissing disease because at one point it was thought that that was how it was spread. But really, mono is spread by just sharing of bodily fluids. So if you drink after someone, it is possible to share mono with them. Mono is a viral infection. It's caused by the Epstein-Barr virus, and it affects lymphocytes. Now let's go back to look at lymphocytes for a moment. Here is a lymphocyte. Now it's hard to tell from this image, but if we look down here at the bottom, it says small, because lymphocytes are very small cells compared to other cells. And when we look at them under a microscope, it's got this very distinct nucleus. But when it is affected by the Epstein-Barr virus, it will cause it to enlarge a bit, and the nucleus becomes misshapen and starts to resemble that of a monocyte. So initially, it was thought that mononucleosis was something to do with monocytes, but it's not. It's causing lymphocytes to resemble monocytes, and that's how it got its name. The lymphocytes have nuclei, or a nucleus, that resembles a monocyte. It's got a mononucleosis. So mononucleosis, lymphocytes infected, resembling monocytes. Okay, now take a breather. We're going to jump ahead and talk just briefly about the immune system. Now, the immune system, in our book, it's a really significant chapter as far as how big it is, and there's a lot of information in it. There is going to be coverage of the immune system when you get to microbiology, if you have to take that class. So that's why we're eliminating a lot of it. We're just cutting it back to some basics because you will be getting it if you need it once you get to microbiology. Now, there are entire master's courses on immunology. There is no way to fit everything in. So when I'm talking about the immune system, I am going to be talking in the vaguest of terms, and make sure to keep in mind that there is actually a lot more going on, even with the parts that we cover in this class. So we are going to be talking about just a small part of the immune system called your adaptive immune system, or adaptive defenses. Our immune system has different branches. There is something called the innate branch, or innate defenses. Those are completely different than adaptive defenses. Adaptive defenses deal with specific pathogens. So something like this year's flu, or we're all familiar with COVID by this point. Remember, there have been different strains of COVID. Well, each strain is a different pathogen. And we would have certain immune defenses against one of those strains and a completely different set against another of those strains. So we are dealing with specific pathogens. But with the adaptive immune system, it is really, really good, except the drawback is we must first be exposed to the pathogen before our body can build a defense against it. Think of it kind of like neighborhood watch. The neighborhood watch could be looking, but if they don't know what to look for, let's say there was a burglar and someone saw the burglar, they can tell the neighborhood watch a description so that the neighborhood watch knows what to look for. But if there's just a person walking through, well, we're not going to respond to him or her, because That's not what we're looking for. We are looking for someone who fits the description of the burglar. Well, that's how your adaptive immune system works. We must first be exposed to the pathogen so that we know what it looks like. What do I mean by what does it look like? Shapes of the antigens. Here's where that's coming back. When we are exposed to a pathogen, really what we're being exposed to are the antigens on its surface. All things have antigens on their surface. Once we're exposed to them, we know what shape those antigens are so that if it shows up again, our immune system recognizes it. Now, when we talk about the adaptive immune system, there's two branches of the adaptive immune system. There is humoral immunity and cell-mediated immunity. The difference in the two, humoral immunity deals with pathogens in our humors. Well, humors is a really, really old term that just means the liquids of the body. But another way to think about that is these are things that are inside of our body, but outside of any cells in our body. So they're in the extracellular environment. That's the humors. The other branch, the cell-mediated immunity, this is immunity concerned with things that have already gotten into our cells. So things like viruses that are inside of a cell. There are a few bacteria that can get inside of your cells. That's less common than viruses. Most bacteria stay outside, but there are some that can get inside the cells. But this also handles things like cancer, because cancer is happening with our cells at the inside level. So that brings us to antigens again. And I really, really hate the way our book handles this word at this point. An antigen, by definition, is any substance that can provoke an immune response. We have, up to this point, talked about antigens being proteins or those carbohydrates on the surface of the cell sometimes, but really it's all about the things on the surface of all things. Now, we have antigens on the surface of our cells. That's what we talked about in blood typing. But remember, antigens are on all things. In this chapter, in chapter 21, Immune System, I think our book really drops the ball, because here our book uses the word antigen the wrong way. Our book uses the word antigen to mean foreign antigen. Remember, your cells have antigens, but so do bacteria, so do viruses, so do parasites. Our book, when it says antigen in chapter 21, where it matters most, it does not mean antigen. It means foreign antigen. And our book uses a completely different term in chapter 21 to mean self-antigen. They are called major histocompatibility complex, or MHC. When you are reading parts of chapter 21 and you see MHC, or major histocompatibility complex, it means self-antigen. And in chapter 21, when you read the word antigen, it means foreign antigen. But antigens in general, are the foundation of how adaptive immunity works. We are going to talk specifically about humoral immunity. Humoral immunity. Now, when we talk immunity, specifically humoral immunity, there's different ways that our body builds this type of immunity. It could be active or passive. The difference in the two is which organism made the antibodies against the pathogen that we're talking about. If You made your own antibodies. That is active humoral immunity. If someone or something else made the antibodies that are in your body protecting you against a pathogen, that is passive humoral immunity. Active, you made the antibodies. Passive, someone or something else made the antibodies. But there are different types of active and there are different types of passive immunity. Let's look at different types of active immunity first. Naturally acquired active immunity. Well, this is what happens when you get sick, or sometimes even when you don't realize it. But when a pathogen enters your body, your immune system will start to make antibodies against it. So you are making the antibodies against a pathogen that has infected you. Another type of active immunity is artificially acquired active immunity. In artificially acquired, here you got a vaccine. When you get a vaccine, you are being injected with either small pieces of a pathogen that cannot harm you, or attenuated, which basically means it's made so that it cannot infect you and cause an issue. So you are being injected with things that have the shape of pathogens, but they cannot harm you. So when you are injected with those, your immune system starts to build antibodies against them. This is artificial because you did not naturally come into contact with those pathogens. They were injected into you, and you started making those antibodies. But in both of those cases, naturally acquired and artificially acquired active immunity, you started making those antibodies. Let's look at passive. There's also a naturally acquired and artificially acquired passive immunity. Passive, we said someone or something else made the antibodies. So let's look at naturally acquired. When baby is in the womb, antibodies from mom can cross the placenta and get into baby's circulatory system. And those antibodies will remain in baby for quite a while, even after he is born. So baby is born with some immunity against whatever mom had immunity against. That is naturally acquired because mom is carrying the baby naturally. If mom breastfeeds the baby after it's born, some of her antibodies are also in the milk that she produces, and those enter the baby and circulate, providing even longer immunity. Again, naturally acquired. Naturally acquired because both of those processes are natural. Passive because it was mom that made the antibodies, and those have been put into baby. So baby is benefiting from them, but baby did not make them. And lastly, artificially acquired passive immunity. This one can be a little tricky to understand. So let's give an example. You're out walking in the woods and you step across a log and you feel something and you look down, and of course you have been bitten by a rattlesnake. Well, that rattlesnake has just injected venom into you. Venom is a protein. It is, in effect, an antigen. So your body is going to start to make antibodies against it. That's naturally acquired active immunity. The problem is most venom acts quicker than your immune system. So it's going to be, probably close to 2 weeks before you build any antibodies against that venom. And by then, you're good and dead. But you get to the hospital quickly and they ask you, what was it that bit you? Well, you can't just say snake. You would need to know, what did the snake look like? We need to know what kind of snake it is. And if you've ever heard someone gets bit by a snake and goes to the doctor, what do? They inject antivenom. Here's what antivenom actually is. They take a specific venom and inject just a tiny little bit into a rabbit or a horse. I don't know why they always choose rabbit or horse, but that's what it is. They inject it into a rabbit or a horse, and that rabbit or horse starts to make antibodies against that venom. And then they do that again a few more times, so there are lots of antibodies against a specific venom. They inject other venom into other rabbits or horses to get other antibodies. Now they take those antibodies, pull them out of the blood, so now they have basically a tube of antibodies against a specific venom. You get to the doctor, they decide, okay, you were bit by this type of snake, let's get the antibodies against that specific snake, inject it. Now you have antibodies in your body to fight against that venom. Well, that is artificially acquired. You were injected with it. It is passive because someone or something else made those antibodies. Another type of artificially acquired is some disorders like certain types of hepatitis or certain types of sexually transmitted infections. Some different disorders, there are people who have been exposed that then start to make antibodies against them, and they can donate those antibodies so that if you believe that you have come into contact with one of those disorders, you can go to the doctor and you can be injected with those antibodies, preventing you, hopefully, from getting infected. Again, artificially acquired because it was injected. Passive because somebody or something else made those antibodies. All right, so let's talk about the different types of cells in the adaptive immune response. They are going to be lymphocytes. There are B lymphocytes, sometimes just called B cells, or T lymphocytes, sometimes called T cells. B lymphocytes are involved in humoral immunity. T lymphocytes are involved in cell-mediated immunity. There is really just one type of B cell. There are other classes, but we just kind of say there's one type of B cell. There are a couple of different types of T cells. There are cytotoxic T cells, and there are helper T cells. Helper T cells really are the guys that say, hey, we've got an infection, we need to do something about it, and it turns the rest of your immune system on. So, helper T cells, we're not really going to talk about cytotoxic T cells, but we are going to talk quite a bit about B cells. So, we keep saying antigens, foreign antigens, but we also said that our body has to come into contact with those antigens for us to have an immune response to it. Well, where does this antigen encounter typically happen? It can happen anywhere in the body, but most often it happens in the lymph nodes. we'll see why it tends to happen in the lymph nodes when we get to blood circulation. But our lymph nodes, when you feel miserable and your joints start to hurt, especially like in your armpits, and then also right underneath your jawline can feel kind of swollen. That's because there are some lymph nodes there. And when our immune system encounters an antigen or a foreign antigen, then our B cells and T cells begin to reproduce. We start to make more of them. If this is happening in your lymph nodes start to fill up with all those excess B cells and T cells. That's why they feel swollen. So let's talk about the activation of your immune cells. When one of your immune cells, those B cells or either types of those T cells, when they come into contact with an antigen that they are specific to, so that's important, the specific to. One B cell is going to be looking for a certain shaped antigen. A different B cell looks for a different shaped antigen. So every antigen will only be able to come into contact with certain B cells or T cells for it to be recognized. When a B cell or a T cell encounters its specific foreign antigen, we're going to go through a couple of processes. First, we're going to go through clonal selection. We start to make clones or copies of that specific activated cell. We will see this in just a moment. We also go through proliferation and differentiation. The differentiation is that once one of these cells, these B cells or these T cells, becomes activated and starts to go through its clonal selection, it's going to become two different types of cells. It's going to become some cells that are active in this current immune response. But it is also going to produce some cells that are not going to do anything at all right now. They're going to go hide in case the same pathogen comes back down the road. We're going to see what those are called in just a moment. But once we produce one of the active cells and one of they're called memory cells, well, those active cells start to proliferate. Those memory cells start to proliferate. So we get lots and lots of effector cells that are ready to fight right now, lots and lots of memory cells ready to fight later. But before we talk about what happens with antibodies, let's do a little bit of drawing. We're ignoring T cells for our class. We are just going to talk about B cells. But B cells and T cells behave similarly. For right now, let's talk about B cells. And across the top, I've got lots and lots of inactive B cells. These cells have not yet come into contact with their specific antigen. We say they are naive. Naive means they're just there. They are not effector cells. They're not ready to fight right now. They're not memory cells. They're not waiting to look for something down the road. They're just there. They're going around and they have this receptor. They actually have several of them, but they have this receptor. And if we look at that receptor, and that receptor, and that receptor, and that receptor, and that receptor, they're all different. They are shaped differently. The one on the left is looking for a round-shaped antigen. The second one is looking for a square-shaped antigen, and on and on. Each one looks for a different shape antigen. And they are constantly going through against every surface in your body, against the surface of every cell in your body. And they are touching those receptors against every antigen in your body. So, for example, let's look at the square guy. Square guy comes up and is going to touch his receptor against this antigen. This green guy down here with the round antigens on his surface, we're going to call him a bacteria. In reality, bacteria are much smaller than any cell in our body. I'm just drawing everything so that we can see it. B cell, with his square-shaped receptor, bumps his receptor up against the round antigens of that bacteria. Square does not fit round. So B cell, what does he do? He ignores him and goes away. It didn't fit. And remember, for a cell to become active, they have to fit. Well, triangle guy comes along. Triangle-shaped B cell comes along, and he bumps his receptor up against the round antigen. It doesn't fit, so the B cell leaves. Well, all of these different shaped B cells of every shape imaginable is going to be coming along and bumping up against every surface in your body, including the pathogen. He's going to touch his receptor to it, and he's going to completely ignore it. But at some point, round-shaped receptor B cell is going to come along, and his receptor fits. And when it fits, we are going to activate the B cell. Now, not super important, because if there is a single bacteria in your body, there are countless copies of that bacteria in your body. So what happens to this one bacteria? Not a big deal, but let's look to see what happens with this one bacteria. This B cell, the very first time he encounters his specific foreign antigen, he's going to rip that antigen off. And this B cell is going to phagocytize. He's going to eat this bacteria. That is not the job of a B cell. That happens one time when he very, very first encounters it. So I'm mentioning it, but you can feel free to ignore it. The one time a B cell phagocytizes is the very first time he comes into contact with his specific foreign antigen. But that's not what we're really concerned with. What we are concerned with is now what happens to this B cell. He's going to take that foreign antigen, find a helper T cell. Remember, helper T cells are the ones that turn our immune system on. And he is going to show that antigen to the helper T cell. So basically what he's saying is, hey, I found this. We need to do something about it. That helper T cell is going to finish the activation process of this B cell. And then the helper T cell is going to leave and turn on all of the rest of our immune system. Don't worry about all the rest of our immune system. We are only covering B cells. So this B cell, what does he do? I'm going to redraw him near the top. This B cell that has already come into contact with his foreign antigen is going to divide. He's going to produce something called a memory cell, and he's going to produce something called a plasma cell. Memory cells divide and divide and divide, producing lots and lots of memory cells. And they go and hide. They hide in your lymph system. Plasma cells are going to divide and divide and divide, but they are going to be active right now. And what they're going to do is start to secrete antibodies. Remember, antibodies, they could be shaped a few different ways, but they're always branched. And at the end of all of those branches are going to be receptors shaped exactly like the original receptor of the B cell. So they are going to be shaped against that round antigen. Now, these antibodies are going to go through our bloodstream. They're going to be out in the tissues of our body. They're going to be in our lymph system. They're going to be all over. And every time they come into contact with that original shaped antigen, which remember was on a bacteria, it's going to stick to it. Well, now we see how we get some sort of immune response when we get an infection. When we're talking about bacterial infections, viral infections that are outside of our cells. If it's inside our cells, there's a whole different process, and we are not concerned with that in our class. But let's go back. Let's look to see, well, we have those antibodies. They're attaching to the bacteria or whatever foreign pathogen it happens to be. What then? Well, there's a few different things. One, there's something called neutralization. Bacteria or viruses or whatever are going to use their antigens to attach to various parts of our body, sometimes getting inside of our cells, sometimes hanging onto the surface of our cells. But neutralization, one of the things that we're doing is covering up those antigens on the surface so that that pathogen can't do those things. As we saw in blood cells, we cause agglutination, but that also works with bacteria or viruses or whatever. We're just going to cause them to all lump together. There's something called precipitation. Well, some antigens are so small that they are soluble in liquid. But if we attach a bunch of antigens to them, a bunch of antibodies to them, they are going to settle out of the blood. And things like our kidneys can remove them, or other phagocytes can come along and grab them. There's something that we're not going to talk about called complement proteins. Well, antigens on the surface get antibodies bound to them, and then a bunch of other proteins in our blood recognize those antibodies and poke holes in the pathogen. We're not going to worry about how that happens. But if we have the neutralization or the agglutination or the precipitation, that can then enhance phagocytosis. It makes it much easier for phagocytes to come along and grab that pathogen and eat it. Those complement proteins can also enhance phagocytosis. It can enhance inflammation. It can cause cells to rupture, lysis. So antibodies themselves, they don't kill anything. They don't harm anything. They just make the thing less harmful, the bacteria or the virus or whatever. They make it less harmful, and they make it so that other parts of our body can kill the pathogen. Now, the first time that our body comes into contact with a pathogen, like what we just drew, well, we start to make antibodies against it. But from the day that we are first going to, let me see if I can circle this, the day that we first come into contact with that pathogen, it's almost a week before we start to make any antibodies, because that process where we're making more and more and more of our immune cells, that takes a while to get started. And then once they do start making copies of themselves, then we can start to make antibodies. But it takes a while before we get to the maximum number of antibodies. All in all, it's about 10 days to 2 weeks before we get that immune response. And during that time, we feel miserable. We're sick. We feel gross. Then after an amount of time, that those antibodies start to kind of taper off. Sometimes it's a little slower, sometimes it's not as drastic, but it does start to taper off. If that same pathogen shows up again at another time, here it shows it showing up 28 days later. It could be months later. It could be years later. But remember those memory cells that went and hid? The thing that kind of justifies them going to hide is that they, even though they were not active in the original fight, they are ready to fight. They do not have to go through this delay. They are ready to immediately start pumping out antibodies. As soon as they are encountered, they start to make antibodies. In this first encounter, we can see not really a lot of antibodies. But here, on this second or later encounter, the antibodies come about immediately and in force. There are a lot of antibodies, and the antibodies stay elevated. So this is why you don't get sick from the same thing a second time. Yes, you've probably had the flu twice. Yes, you have had strep throat twice. But when that happens, it's not the same strain. You are immune to something once you have come into contact with it, because every time that you come into contact with it after, this happens. And you don't even realize it. You don't get sick. You don't feel miserable because it is so exploded that second time that you come into contact with it and later. So that's tied into vaccines, and everyone has some really strong opinions about vaccines. So what do we think? Vaccines? Yes? No? And defend your reasons. Why do we get vaccines, or why shouldn't we get vaccines? Especially right now, shortly after COVID, there was a whole bunch of really strong opinions then. So let's take a look to see what's going on with vaccines. When we get vaccinated, as we saw back here with this, we are injecting someone with either dead viruses, dead bacteria, attenuated, which just means they are alive, but all of the harmful stuff has been removed, or even pieces of viruses or bacteria, just the antigens. So when we do that, what happens? Well, that's what we drew here. We inject it, and then we go through this process. We make plasma cells to create antibodies. We make memory cells. These are the important part. These, now, whatever you got vaccinated against, later, if it shows up, what happens? this second branch happens. But at the same time, if you get a different strain of flu, so every year they make the flu shot, and some years they guess right. Some years they guess the wrong strain. They do their best, but maybe they go against H1N2 and instead H5N3 shows up. Well, in that case, it's just like your first exposure to any pathogen. You have that delay, you feel miserable. So you got your flu shot and it just didn't work. But if you got your flu shot and then that strain of flu shows up, now we get this and you don't even realize that you were exposed to the flu. So that's how vaccines work. But does that tell us yes or no? Well, everyone has heard about the link between vaccines and autism or vaccines and other disorders. That is all because Andrew Wakefield, back in the, I believe, early to mid 80s. What does this say? Yeah. 1990 something. So early 90s, this guy, Andrew Wakefield, writes this report. And there's a lot going on, and he never actually mentions the word autism. That wasn't really a big term back in the 90s. It was just starting to come into common speech. But Andrew Wakefield, in this, basically what he points out is that when people are given vaccines, there is a higher rate of illnesses, including autism, although he never says autism. So there's this link between people who get vaccines and autism. And of course, when that shows up in the Lancet is a very prestigious medical journal. So very prestigious medical journal shows that this man demonstrated a link between vaccines and autism. That is pretty scary because everyone was getting vaccines. But if you look up the article now, this is what you find. Because when they looked a little more into it, what they found out was Andrew Wakefield faked the entire study. He paid some people to say some things that were not true. He invented some data that was not true, and he was found out. And what they discovered was, no, there was no link between vaccines and autism. The problem is the damage was already done because everyone hears about vaccines causing autism. No one hears about the paper that was retracted and made up. But that's just one part of the argument. Let's talk about the whole vaccines and autism nonsense, or the vaccines and other things that they say that it causes nonsense. Well, this woman, famous for eating her boogers on national television, says that vaccines cause autism because her child got autism right after getting vaccines. How can you not believe a face like that? Well, let's see what the actual studies say. People say there's mercury in vaccines. Well, there is something called thimerosal in vaccines, and thimerosal contains mercury. What do we know about mercury? Mercury is a heavy metal. It causes all sorts of mental and muscular disorders. So how is it safe to be in vaccines? Because thimerosal is in vaccines, or at least it was. So mercury was in vaccines. The thing is, there are different types of mercury. Sodium is deadly. If you eat sodium, it will kill you. Chlorine is deadly. If you breathe in chlorine, it will kill you. But every single day, you eat both of them. Because when sodium and chlorine come together, they make table salt. And once you get that table salt in your body, it's broken apart into sodium ion and chloride ion, and both of them are harmless. Well, mercury is the same. There are different types of mercury, and the mercury that was in thimerosal It's harmless. It does not hurt you. But on top of that, thimerosal was removed from vaccines way early on, late 70s, early 80s, just because people were afraid of mercury. There's only 1 or 2 vaccines that have thimerosal in them now, and they are hardly ever given. Next, formaldehyde. Formaldehyde is what they preserve dead bodies with. How can that be safe? It's in vaccines. Yeah, it is absolutely in vaccines. It's a preservative. But the amount of formaldehyde that's in a vaccine is less than the amount that your body naturally produces when you eat a meal. And it lasts for a shorter period of time. Formaldehyde, yeah, it can be deadly, but it doesn't necessarily have to be. It's in a very small amount, much smaller than your body produces every time you eat. Antifreeze. There is antifreeze in these vaccines. Yeah, there absolutely is. These vaccines are typically stored at very, very cold temperatures, far below freezing. So we put something in there to keep it from freezing. But it's not the same antifreeze that you put in your car. This is a natural antifreeze. Most fish produce antifreeze to keep them alive when they're in cold water. And we eat fish all the time, and it doesn't hurt us. The antifreeze that is in vaccines, similar. Doesn't hurt. It's just there to keep it from freezing when it's in these cold temperatures. What about the timing? We start to give these vaccines at a really young age, and we start to see more and more and more young people getting diagnosed with autism at these really young age, sometimes just a few weeks after getting these vaccines. So the more vaccines there are, we start to see more autism. There has to be a link, right? No. We're giving more vaccines because we're able to give more vaccines. The reason that the timing seems so suspicious is we're getting really good at diagnosing autism really early. So early, in fact, that we can usually diagnose it right around the time that vaccines are given the first time. There is no link. The timing just happens to be laid out in that way. Well, so what? Let's say, for the sake of argument, that vaccines did cause autism. Is autism really worse than paralysis that comes from polio, death that comes from things like measles or smallpox? No, it's not. Even if vaccines caused autism, which they don't, autism is preferable to a dead baby. Well, what about people that say, well, I got the vaccine and I still got the flu? Well, like I said a minute ago, we're not always 100% positive when we're coming up with what flu is going to come through this year. We need to make a vaccine before it gets here. Or I got the vaccine and it gave me the flu. No, it didn't. Sometimes you get the vaccine and a few days later, you get whatever it was you got vaccinated against. That does not mean that that gave it to you. It means that you already had it because it's a few days before you start to show symptoms. When you start to show symptoms, you actually got it a few days before. So what's happened is you went and got a vaccine, yay, but you didn't get it early enough. You already had whatever it was you got vaccinated against. And then you start to show symptoms. So let's look to see what happens with rates of disease before and after introduction of their vaccine. Diphtheria, before vaccination was invented, after vaccination was invented. 21,000 something cases a year to 0. That is a reduction 100% because of vaccines. Influenza, 99% reduction. Hepatitis A, 91% reduction, on and on and on. Polio, 100% eradicated after its vaccine was introduced. Smallpox was the first disease to be completely eradicated because of vaccines. All of these things, and these numbers may have even gotten better because this is a few years old now, but all of these things, look at that dramatic reduction right after the vaccine was invented for it. So yeah, the answer, vaccines, yes. All positives, no negatives. There's no reason not to get a vaccine. It's not going to hurt you. And to sum it up, vaccines cause adults. There is no reason to not get your child vaccinated. Help them to be healthy for the rest of their life. So let's have a thinking cap question. Explain the following statement. You have had a cold more than once. You've probably had strep more than once. You've probably had the flu more than once. But you have never had the same cold, the same strep, or the same flu more than once. Explain that. Well, we kind of already did a little bit ago. Yeah, you've had more than once exposure to the cold, and you've gotten a cold from it. But every time you get a cold, it's a different strain. Every time you get the flu, it's a different strain. Every time you get strep throat, it's a different strain. Because once you get one of those, you are immune to that strain because of antibodies, because of memory cells. Those two things, antibodies and memory cells, keep you from getting the same thing twice. All right, that's how the immune system works. So we will wrap up there. We've got one more lecture in this block, one more lecture in this chapter. We will talk about platelets in our next and final lecture for this section. All right, everybody take care, and I will talk to you next time.